All right, let's dive right into this high-yield medical virology explainer, designed specifically to give you the mental cheat codes you need for your board exams. Now, we know virology can feel like just an endless list of microscopic details, but by understanding a few core structural and genetic rules, you can literally logic your way through almost any question they throw at you. Here's the game plan for today. First, we'll cover structure, specifically envelopes and capsids. Then, we'll decode RNA viral genomes, move on to DNA viruses and retroviruses, and finally, tackle shift, drift, and host defenses. Let's get to it.
Starting off with Section 1, Structure, Envelopes and Capsids.
Think of the nucleocapsid as the core of the virus. Now, if a virus wraps itself in an outer lipid layer, you'd probably think, wow, extra armor, right? Actually, scratch that. It's a massive weakness. Enveloped viruses like HIV, the flu, or hepatitis B are surprisingly fragile. A little bit of everyday soap and water or alcohol literally tears that lipid membrane apart. But non-enveloped or naked viruses, those are the absolute tanks of the microbial world. Because they just have a dense protein shell, they laugh at detergents and bile salts, which is exactly how nasty bugs like norovirus or polio survive the brutal gauntlet of your GI tract. To kill those on a surface, you're going to need the heavy hitters like bleach or peroxide. Now, inside that envelope, you absolutely have to know your capsid symmetry. Most of the time, you're looking at icosahedral shapes. You know, those perfect 20-sided geometric dice. Others are helical, kind of like a spiral. Think influenza, rabies, or measles. But watch out for pox viruses. They are the ultimate rule breakers. They don't fit into either of those neat little boxes. They have a totally complex structure all to themselves.
Moving right along to Section 2, Decoding RNA viral genomes.
Okay, this is an absolutely crucial concept. If a board question hands you a positive strand RNA virus, just think of it as ready-to-go mRNA. The second it breaches the host cell, boom, it translates directly into proteins. No special enzymes needed, it just goes to work. But negative strand viruses? They're basically helpless on their own. They're RNA a mirror image, a complement to mRNA. So if they don't pack their own special enzymes, specifically a virion-associated RNA-dependent RNA polymerase or transcriptase, they literally cannot replicate. They are dead on arrival without it.
So, putting the RNA rulebook into practice, there's a massive high-yield association you've got to memorize. Notice a trend with those scary negative strand viruses like influenza, rabies, Ebola, and Marburg? Every single one of them happens to be enveloped. Always. Meanwhile, positive strand viruses like corona or flaviviruses can be either enveloped or naked. And then there's the double-stranded RNA viruses, like rotavirus, which are non-enveloped but notably carry a segmented genome.
Speaking of rotavirus, I want you to burn this number into your brain. 11. Rotavirus has exactly 11 distinct segments of double-stranded RNA. Why does that matter? Because having a highly segmented genome like a chopped-up puzzle is a massive setup for reassortment. It's a total game-changer for viral evolution and, for sure, a very frequent exam target.
All right, on to Section 3, DNA viruses and retroviruses.
Now, with DNA viruses, most of them are double-stranded. Heavyweights like herpes, adenovirus, and HPV, they all follow the rules and travel straight to the host's nucleus to replicate. Simple enough, right? But then you have the single-stranded outlier, parvovirus. Parvovirus B19 is highly tested because it triggers some very unique clinical presentations, most notably causing an aplastic crisis in patients who have sickle cell disease. But you know how the boards work, right? They absolutely love testing the exceptions. So, ask yourself, which DNA virus flat out refuses to go to the nucleus to do its business? Yep, it's our old friends, the pox viruses. Like we mentioned earlier, these guys are the ultimate rule breakers. They are massive, complex, double-stranded DNA viruses that literally bring their own construction site with them. Because they pack their own DNA-dependent RNA polymerase, they don't need the host's nucleus at all. They're the only DNA viruses that replicate exclusively in the cytoplasm. Let's pivot and look at a totally different beast, the retroviruses, like HIV and HTLV. These guys start out as positive strand RNA, but instead of acting like normal mRNA, they pull a fast one. They use an enzyme called reverse transcriptase to run the whole process backward, turning their RNA into double-stranded DNA. And here's the truly terrifying part, step 3. That newly minted DNA integrates permanently into the host's own chromosome as a provirus. Your own enzymes transcribe it. It essentially becomes a permanent, hardwired part of the infected cell.
And that brings us to our final section, Section 4, Shift, Drift, and Defenses. Okay, you are 100% going to see a question on the difference between shift and drift. Let's look at antigenic drift first. It's slow. It's that gradual, sneaky accumulation of point mutations over time. It's exactly why we get seasonal flu epidemics and why we all need a new flu shot every single year. But antigenic shift, that is a violent, abrupt, major overhaul. And remember, it only happens in viruses that have segmented genomes, like influenza or rotavirus. When two different strains co-infect the exact same cell, they swap out entire genome segments. The result? A totally new, highly lethal reassortment. Simply put, drift causes epidemics, but shift causes pandemics.
All right, let's hit some rapid-fire vocabulary that's absolute gold for the boards. First up, CPE, or cytopathic effect. Think of this as the visual footprint of destruction a virus leaves behind. When you peer through a microscope and see cells inappropriately rounding up, bursting open, or forming these weird inclusion bodies, you're literally looking at the structural carnage caused by the virus hijacking the host machinery. Next is latency. You'll see this tested a ton, especially with herpes viruses. Latency just means the virus is hanging out in the host, totally chilling, not actively making infectious progeny. It essentially goes to sleep in your tissues. Herpes simplex virus, for example, is famous for hiding out in your dorsal root ganglia neurons, just quietly maintaining the ominous ability to wake up and reactivate years or even decades down the line. Now, what if an exam question asks you to actually quantify how infectious a virus is? You'll likely see the term TCID50. That stands for Tissue Culture Infectious Dose 50%. It's the exact, precise concentration of a virus you need to successfully infect exactly half of the cell cultures you've inoculated. It's the absolute standard benchmark for measuring viral potency. Alternatively, they might throw PFU at you, which is plaque-forming units. This is more of a direct head count of infectious viral particles. Imagine spreading a virus over a nice flat layer of cells. Wherever a single infectious virus lands and starts wrecking shop, it creates a clearing, a dead zone called a plaque. By literally counting those plaques, you can calculate the exact viral concentration based on those areas of cytopathic effect. Of course, our bodies don't just sit back and take the hit. We fight back, immediately, using our innate antiviral immunity. This is primarily led by type 1 interferons, alpha and beta. And this whole defense system is tripped the literal second our innate receptors spot foreign viral nucleic acids floating around where they have absolutely no business being. Whether it's single-stranded RNA triggering TLR7 and 8, double-stranded RNA hitting TLR3 or RIG-I, or just rogue viral DNA activating the CGAS receptor, the host cell instantly sounds the alarm to shut down viral replication. So I want to leave you with a final thought to tie all of this together. We know that the reassortment of segmented genomes is basically the ultimate cheat code for viruses to bypass our immune system. If a single host cell gets simultaneously invaded by both a human and an avian influenza strain, those RNA segments can perfectly shuffle. So what animal co-infection do you think will be the mixing vessel that triggers the next great global shift? Keep that in the back of your mind as you study these structures, because understanding these virology rules doesn't just help you logic your way through exams, it might just help predict the next pandemic.